Hi everyone, this is the second recording of the chapter recombination of paper 2. In the previous recording, we have seen what is recombination, what is its significance, and the Holliday model for homologous recombination. In this recording, we are going to see another model for homologous recombination, which is the double strand break repair model or double stranded break repair model for homologous recombination. Homologous recombination is often initiated by double-stranded breaks in the DNA. So a common model describing this type of genetic exchange is a double-stranded break repair pathway. As in Holliday model, here also the first step is alignment of homologous chromosomes. As we have seen in the previous recording that homologous chromosomes are almost or nearly identical with each other, but despite this high degree of similarity, there is some difference in the sequence. And in these two homologous chromosomes on the slide, one highlighted in gray, another shown in blue color for explanation purpose. The one highlighted in gray has two genes, capital A and capital B, and the one highlighted in blue has again two genes, small a, small b. These are the alleles, capital A, small a, and capital B, small b. That is different forms of the gene. Now, they will be almost identical, but let's say there are some sequence differences in there. So, in contrast to the Holliday model for homologous recombination, where you have seen in the previous recording that a single-stranded nick takes place at the identical position in both the DNA molecules, here a single-stranded nick doesn't take place. Instead, there is a double-stranded break, also called as DSB. That's why it's called as double-strand break repair model. There's a double-stranded break, and the twist is, again in contrast to the Holliday model, where the single-stranded nick takes place and here a double-stranded break takes place. The another difference is that in Holliday, as per the Holliday model, the single-stranded nick takes place in both the DNA molecules, that also at the same position. Here, the double-stranded break takes place only in one of the two DNA molecules. So in this case, what we have seen that the double-stranded break takes place in this DNA molecule. Whereas the other DNA molecule, there is no break. The other DNA duplex remains intact. And because the double-stranded DNA breaks occur relatively frequently in cells, this type of event is attractive compared to the nicks that are proposed to initiate recombination by the Holliday model. However, this asymmetric initial breakage of the two DNA molecules in the DSB repair model necessitates that the later stages in the recombination process are also asymmetric. This double-strand break repair model is very accurate. Now, after there is a double-stranded break in one of the DNA molecules and the other DNA remains intact, what happens next? What happens next is there is processing of the broken DNA molecule to generate DNA with single-stranded tails. So you can see the break which has taken place, double-stranded break in one of the DNA molecules in this first step, generates blunt DNA ends. You know the difference between a blunt DNA end or a sticky ends. So this blunt DNA end has to be processed. So DNA processing takes place at this broken DNA ends to generate single-stranded DNA tails. So after the introduction of this DSB, a DNA cleaving enzyme, which in the future recordings we will see which enzyme is this, a DNA cleaving enzyme sequentially degrades the broken DNA molecule to generate regions of single-stranded DNA. So you can see in the next step, after this broken DNA is processed, you have regions of single-stranded DNA. These regions or extensions are called as ssDNA tails, and these tails terminate with 3' end, which is very important. Then what happens next? A double-stranded break took place in one of the DNA molecules, the other one remained intact. Then the processing took place to generate DNA with single-stranded tails with 3' ends. What happens next? The next step would be strand invasion, which you have seen in the Holliday model as well. So this single strand tail, this single stranded DNA tail, sorry, this single stranded DNA tail, let's say, invades the unbroken homologous DNA duplex. Which one is unbroken homologous DNA duplex? The one shown in blue in the picture, which is unbroken or intact. So this single stranded tail is going to invade this unbroken homologous DNA duplex. In the next step, you can see. So the single stranded tail is invading the unbroken homologous duplex and it's pairing with its complementary strand in this other duplex DNA. So if I say that, or if I use this language, that the single stranded tail is highlighted gray in color, whereas the unbroken DNA, both the strands are blue in color. So some melting takes place where the blue strands get separated from each other or peeled away, and whereas the gray strand, the invading strand, pairs with the blue strand. This is the strand invasion, which we have seen in the Holliday model as well. Now, what about if the other blue strand, which was peeled away from its parental strand, it's gonna pair with the other gray strand, which is this one. So you can see in the next step, the second strand invasion takes place where this blue strand, which was separated from its parental strand, it's pairing with this gray strand. So this is the second strand invasion. And now the repair synthesis would take place, and elongation from this DNA ends using the complementary strand in the homologous duplex as a template serves to regenerate the regions of the DNA that were destroyed during the processing of the strands at the break site. So if you say that in the first step, when the double-stranded break was generated, and this processing took place to generate this single-stranded 3' tails, some DNA was degraded here to generate these tails. Yes, this degraded DNA will be repaired now. So the DNA which was destroyed during the processing of strands at the break site will be regenerated. After the repairing, you can see there are two X-shaped structures which are the Holliday junctions. We have seen a Holliday junction in the Holliday model as well, but there was one Holliday junction as per the Holliday model. In the double-stranded break repair model or DSB repair model, there are two Holliday junctions. And now branch migration would take place. These Holliday junctions are gonna move. Then we come to the last step in the recombination. So whether it is Holliday model or whether it is double strand break repair model, the first step is alignment. Then there is nick in the DNA, which is a nick in the one of the strands, and the both DNA molecules at the same position in Holliday model. Whereas in DSB repair model, it's a double-stranded break in one of the DNA molecules where the other one remains intact. Then strand invasion takes place, and then branch migration, and finally the resolution will take place. Before I talk about resolution, you also need to remember that in DSB repair model, there is processing also after the generation of the double-stranded break. Now, there are two Holliday junctions here. Let's consider or call them as junction X and junction Y. Now, at both the junctions, you can see there is site 1. And you can see the corresponding arrows, and there is site 2. So at both the junction, there is site 1 and there is site 2. Now, where the cleavage will take place, that will decide whether the products would be patch or the non-crossover products, as we have seen in the Holliday model as well, or splice or crossover products. So whether there will be, reassortment of genes or no, that will be decided on the basis of where the cuts would be. So if the cleavage occurs at the same site at both the junctions. When I say cleavage occurs at the same site at both the junctions, so let's say if both junctions are cleaved in the same way, that is at either both at site 1 or both at site 2, then non-crossover products will be generated. So if the junction X is cleaved at site 2, which is the horizontal cleavage if you see these arrows, and the junction Y is also cleaved at site 2, which is the horizontal cleavage. Again, you see the arrow. So if both the junctions are cleaved at site 2, then you have non-crossover products. So you can see here resolution at site 2 in both X and Y as I have drawn. So you have non-crossover products or the patch products, and no reassortment has taken place, because you can see with the capital A, there is capital B only. And with the small a gene on this DNA, there is small b only. So no reassortment, non-crossover, non-recombinant or patch products, because the resolution has taken place at the same site at both the junctions X and Y, which is site 2. Now, if the resolution takes place at site 1, which is your vertical cleavage, see the arrows, at junction X, and at junction Y also it is site 1, again vertical cleavage. So again, you're going to have the non-crossover products only, which is the patch products, no reassortment. With capital A, you will have capital B, and small a, you will have small b, because both the junctions are cleaved at the same site, that is site 1. So if both junctions are cleaved in the same way, that is both at site 1 or both at site 2, then non-crossover products will be generated. Okay. Then what will generate the splice products? Now, to generate splice products, resolution or the cleavage should take place at different sites. If the holiday junctions are cleaved using different sites, then the crossover products will be generated. Now, if the junction X is cleaved at site 1, which is this vertical cuts, and junction Y at site 2. So your junction Y at site 2, which is your horizontal cuts, then you have splice or the crossover products. And if you see here, in this DNA molecule with small a, you have capital B gene, and with capital A, you have small b gene. So this is reassortment. These are crossover or splice products, and recombination has taken place. And, or I would say recombinant products are generated because the junctions are cleaved at different sites. Site 1 for junction X and site 2 for junction Y. Now, if the junction X is cleaved at site 2 and Y is cleaved at site 1, still they are different sites, and you're going to see crossover products there as well. So in this recording, we have seen the double strand break repair model, how it's more accurate model, and the steps in this model, that the two homologous chromosomes align, then a double-stranded break occurs in one of the DNA molecules with the other one remaining intact. Then at the broken end, processing takes place to generate DNA with single-stranded tails ending with 3'. Then how the single-stranded tail invades the unbroken DNA molecule, strand invasion, and there is second strand invasion as well. Repair takes place, resulting in 2 holiday junctions. Branch migration takes place, and then the resolution, depending on whether the resolution at both the junctions are at the same sites or different sites, non-crossover or crossover products are generated. So this is all about double strand break repair model for homologous recombination.